Hello, I am Anna Lisowska, and today I will present my master's research project supervised by Dr. Islem Rekik, which was published in the Journal of Brain Connectivity. It's titled Joint Pairing and Structured Mapping of Convolutional Brain Morphological Multiplexes for Early Dementia Diagnosis. Early mild cognitive impairment, or EMCI, manifests itself as a cognitive decline greater than that expected of person's age and educational level. Since it might progress into Alzheimer's disease, it is important to diagnose it for early intervention. Research looking into changes in the brain's morphology in MCI patients found evidence of cortical thinning, reduced hippocampal volume, and sulcal widening. However, spotting these changes at the very early stages of MCI remains challenging. Although dementia has been shown to affect neuronal connections in the brain as well as the cortical surface, most existing connectomic research focuses on structural and functional connectivity, which may be noisy and difficult to acquire, while overlooking the morphological connectivity. Further, a number of studies reported changes in cortical morphology in MCI patients. However, none of them considered how different brain regions get affected by MCI in relation to each other. To address these limitations in our previous work, we proposed to use morphological cortical networks for dementia onset identification. Additionally to using one layer network, considering only one morphological view such as cortical thickness, we constructed a multi-layer network, or a multiplex, consisting of multiple morphological views. Previous research showed that using multi-layer networks improved the prediction accuracy for disease identification when compared to using single-view networks. However, to the best of our knowledge, no previous methods explored the similarity between layers in a multi-layer network for modeling brain connectivity. Hence, we introduced convolutional interlayers into a multiplex structure to capture the relationship between different brain views. Basically, each multiplex consists of different morphological views, or intralayers, and interlayers slipped between two consecutive intralayers, thereby quantifying the relationship between them. To be able to make use of unique information captured in each multiplex for EMCI identification, we needed a feature fusion method which would capture the most informative features from multiplexes, while minimizing the modality-specific noise. However, most existing network fusion methods often extract features independently from each network and then simply concatenate them into a long feature vector for classification, while overlooking the correlation between them. To address this issue, we propose to use canonical correlation analysis, or CCA, to map two sets of features into a shared space where they become more comparable. CCA was shown to yield more discriminative features than any of the input feature vectors alone or their simple concatenation. Since we were not restricted to only two sets of features, we proposed a novel pairing-based CCA mapping of multiple sets of brain multiplexes where each pair of multiplex sets was mapped onto a CCA space, then fused. Ultimately, in the spirit of ensemble classifier learning, the fused multiplex features were used to train a linear classifier in each spanned CCA space. However, from our previous work, we noticed that using deep multiplex structure with overlapping cortical brain views resulted in many highly correlated features used for the ensemble learning, which may adversely affect the model's performance. To minimize the correlation between different multiplexes, we propose a shallow multiplex structure composed of a pair of morphological brain networks, where between the two networks we slide a convolutional interlayer modeling the relationship between them. Furthermore, since the multiplex data is highly dimensional, we could not apply a CCA mapping to our multiplex sets without previously reducing their dimensionality, specifically using a principal component analysis, which resulted in loss of model interpretability. It was impossible to identify the most meaningful features. This could be remedied by using sparse CCA, which performs feature selection by imposing a sparsity constraint on the linear coefficients. However, One of the limitations of sparse CCA is that it follows an assumption that all the features within the same view are independent of one another, which is hardly the case in brain network data. A variety of methods exist which impose sparsity in a structure-aware manner to capture the high-level structured information, which is of great importance in the case of our data. We chose to use graph-guided pairwise group lasso sparse model introduced by Du et al. 2017 due to the following advantages. First, it can recover the structure information from the input data in an unsupervised manner and without any a priori knowledge. Second, it has a strong upper bound for the grouping effect of correlated variables independently of sample correlation sign. Third, it can find stronger, more stable canonical correlations and cleaner canonical loading patterns comparing to other state-of-the-art SSCCA methods. Also, it can be applied to the network data without the loss of feature interpretability. These were the motivations of our work. I will now provide more details of the steps we took to build the framework for early dementia diagnosis. First of all, for each subject, we construct a single view network representing one cortical attribute, such as sulcal curvature. A single view network comprises a set of nodes or anatomical brain regions, and a collection of edges interconnecting the nodes, representing the dissimilarity between the two brain regions in morphology. The average value of a cortical attribute was calculated for each anatomical region of interest. For each cortical attribute, the strength of each network edge connecting two regions of interest is then computed as the absolute difference between their average values, thereby quantifying their dissimilarity. The same procedure was followed to obtain the connectivity matrices from different cortical attributes, such as sulcal depth or cortical thickness. However, we also want to model the relationship between the two different morphological networks and see how it might get affected by the early MCI. To do this, we construct a higher-order morphological network, or multiplex. We define a shallow multiplex using two morphological brain networks, here maximal principal curvature and mean sulcal depth, where between the two networks we slide a convolutional interlayer which models the relationship between them. More specifically, the convolution blends two networks together and is the expression of the amount of overlap of one network as it is shifted over another. The multiplex architecture allows us to explore not only how different brain views get altered by a specific disorder, but also how the relationship might get affected. We note that each subject-specific brain multiplex captures unique similarities between two different morphological brain networks that are not present in different shallow multiplex. To make use of all the information contained in different multiplexes, we propose to perform pairwise fusion of features from different morphological multiplexes. This strategy requires an ensemble framework, which would allow for the exploration of all possible combinations of pairs of multiplexes. We have n different multiplex sets, each computing unique relationship between different cortical attributes. Due to high dimensionality of multiplex structure, we first need to use principal component analysis, PCA, to reduce the dimensionality of each training multiplex. Then we apply the structure-constrained sparse canonical correlation analysis to map a pair of PCA projected multiplex sets onto a common space where pairs of multiplexes are most correlated. This is repeated for all possible combinations of sets of brain multiplexes. Then we fuse the selected highly correlated features given highest canonical weights from paired shallow multiplexes to train a linear support vector machine classifier. Therefore, we have k classifiers, each taking a different set of fused features as input. The decisions from all the individual classifiers in the ensemble are combined for the final classification decision. In the test stage, the same steps are followed for the testing subject. First, the multiplexes are projected onto PCA space learned in the training step. Then, sets of multiplexes are paired, mapped, and fused using canonical weights assigned during training. and imputed in the ensemble of linear SVM classifiers, which produces a final early MCI versus normal controls classification decision based on a weighted voting from all the SVMs in the ensemble. In our experiments, we used 42 early MCI and 42 normal control, age and gender mapped subjects from ADNIGO public dataset, each with a structural MRI image. We used free circle analysis suite to reconstruct both right and left cortical hemispheres for each subject from the structural MRI images. Then we parcellated each cortical hemisphere into 35 cortical regions using Desikan Kilani atlas, and constructed the morphological networks for the 4 different cortical attributes: the maximum principal curvature, the mean cortical thickness, the mean sulcal depth, and the mean of average curvature. To evaluate the performance of our Ensemble SCCA pairs classifier, we used 4 kinds of features as input into our framework: features from single brain networks, features from correlational 4-layer multiplexes, where the interlayer is computing using Pearson correlation, features from convolutional 4-layer multiplexes, where the interlayer is computing using 2D convolution, and features from shallow 2-layer convolutional multiplexes, which is our proposed structure. We compared the performance of our proposed framework with the following classifier structures: Ensemble classifier, which does not perform any pairing or mapping, but combines information from different views or multiplexes just on decision level. Ensemble paired classifier, which pairs the views or multiplexes, but instead of mapping, simply concatenates them. Ensemble CCA paired classifier, proposed in our previous work, which maps pairs of view or multiplexes onto a CCA space. And our proposed graph-guided lasso SCCA classifier. We test the classification performance of these frameworks on each input feature type separately. We note that the best accuracy was always obtained when using the proposed shallow convolutional multiplexes. Our proposed Ensemble GGL SCCA paired classifier framework outperformed all comparison methods when using shallow convolutional multiplexes of the left hemisphere. In contrast, the best classification result for the right hemisphere was obtained using Ensemble CCA paired classifier introduced in Lisowska and Rekik 2017. This might indicate that the group effect introduced by GGL SCCA may better capture a morphological connectional structure in the left hemisphere that is not prevalent in the right hemisphere. It might also imply different levels of complexity in morphological cortical disease progression across both hemispheres. We employ an ensemble framework to combine information from different pairs of brain multiplexes at the decision level. Each pair of multiplexes is projected onto a structure-sparse CCA space and communicated into a linear SVM classifier. The following graph compares the accuracies of early MCI versus normal control classification obtained by the ensemble framework and each individual classifier that constitutes it. The numbers on the x-axis represent different combinations of brain multiplexes. From this figure, we see that the ensemble framework performed better than any of its symbol classifiers in both left and right hemispheres. It may indicate that different pairs of mapped multiplexes contain complementary information that is meaningful for the early dementia diagnosis. For feature identification, we performed the structure-sparse CCA mapping directly on the pairs of views, as it did not require previous application of dimensional auto-reduction technique, preserving feature interpretability. We identified features that were consistently given the highest canonical weights across the subject and across the pairs of views. Circular graphs are plotted for the top 10 features identified by the structure-sparse CCA for each cortical attribute. The connection given highest canonical weight in the maximum principal curvature brain view was the connection between the transverse temporal cortex and the pericalcarine cortex, which seems to act as a hub region for many highly correlated connections. This is consistent for both cortical hemispheres. Additionally, the transverse temporal cortex seems to serve as a hub region in the right hemisphere. In case of the mean cortical thickness brain view, many highly correlated connections were found. and were concentrated between the frontal pole, the temporal pole, the insular cortex, the bank of superior temporal sulcus, the corpus callosum, and the entorhinal cortex, with these likely being important hub regions. For the mean sulcal depth brain view, the strongest connection was between the entorhinal cortex and the insular cortex, which is identified as a hub region with many connections extending from it in the left and right hemisphere. On the other hand, in the mean of average curvature brain view, the main connection was reaching from the entorhinal cortex to the pericalcarine cortex, which acts as a hub for connections, especially in the left hemisphere, while the entorhinal cortex is also a hub in the right hemisphere. Generally, with the exception of the mean cortical thickness, one very strongly correlated connection was present. The circular graphs provide us with a useful information about the underlying network structure for different cortical attributes. To identify which morphological brain connections may serve as important biomarker for early dementia diagnosis, we first determine which pairs of brain views yield the best early MCI versus normal control classification accuracy. We note that the highest accuracy of early MCI versus normal control classification using cortical attributes from the left hemisphere was obtained by a classifier trained on pair of views consisting of maximal principal curvature and the mean sulcal depth. Interestingly, the pairs which contained the maximal principal curvature as one of the views consistently performed best in case of the left hemisphere, which shows that this cortical attribute provides important insight into dementia progression. For the right hemisphere, the best classification performance was also achieved when using the sulcal depth and the geometric properties of the sulci, specifically the mean of average curvature. These results suggest that the curvature of the sulci is likely affected by the early dementia and could provide insight into the progression of the disease. Based on this information, the most notable connections were found between the entorhinal cortex and the pericalcarine cortex, and the entorhinal cortex and the insular cortex. The insular cortex is involved in emotion processing and regulation, introspective awareness, and integration of multimodal inputs. The entorhinal cortex plays an important role in navigation and formation and consolidation of spatial and declarative memory. Whereas changes in insular and entorhinal cortex prior to the onset of Alzheimer's disease symptoms were identified by previous research, our findings further suggest the pericalcarine cortex, responsible for integration of visual stimuli, as a candidate biomarker for early dementia diagnosis. In this work, we introduced a shallow morphological brain multiplex structure which contains information on the morphological connections between different brain regions, as well as the relationship between different morphological attributes. We then proposed a paired graph-guided lasso SCCA ensemble classifier to make use of features from different multiplexes by taking into consideration the underlying structure information. Finally, we evaluated the proposed framework on the early MCI patients from ADNI-GO dataset. As a result, we observed correlated brain changes in sulcal depth and curvature in early dementia. We are also able to identify insular cortex, entorhinal cortex, and pericalcarine cortex as biomarkers for early MCI diagnosis. Nevertheless, there are some limitations of our work that could be addressed by the future research. For example, our structure-aware sparse CCA assumes linear relationship among variables. However, other methods exist that are capable of modeling more complex nonlinear relationships, such as deep CCA, which learns complex transformations of attributes through deep networks. Additionally, we were restricted to pairwise CCA mapping. However, some novel methods, such as multi-view discriminant analysis, are able to find a common space jointly for multiple attributes. Further, since our mapping strategy is unsupervised, it does not ensure class separation. On the other hand, supervised multi-view CCA includes class label information as an attribute to guide the CCA mapping. Lastly, we restricted our investigation to different morphological attributes, whereas combining these with structural and functional networks and researching the relationship between them would provide a more complete view on the manifestation of early MCI. Thank you for your attention.
For more, please visit us at basira-lab.com and don't forget to subscribe, like, share, and cite our work.